wouldn't give you a final exam if I had to do that. So, no. But again, the final exam is extremely general information. Okay, and honestly, my students historically do very well on it.
Well, it's funny that you say that because last year I finally got my first C on the test and it was on the final, and I never the whole year I was like I'm nuts, and you know I was so excited I got a B. I didn't even know what a B looked like.
See. So, yeah, they do tend to do very well. So I want you to keep that in mind. And don't stress over that. No stressing allowed. Yeah, that's what I thought. It's like, okay, so we talked a little bit about nutrition. We picked on everybody who was eating whatever they were eating in class on Thursday. And I want to share with you my little gadget. Do you know what this is? It's called the jawbone. And what it does is it monitors your physical activity. It monitors your sleep. And then you can hook it up with any of the like the MyFitnessPal we were talking about. And I use that to put my food in, so you can hook it all up together.
Really? Where'd you get that?
I ordered it online because I explained my issue. I was having problems. I can't lose weight to save my life, so I want to make sure that I'm doing everything I'm supposed to do, and then I'll take the next step, like go to the doctors and find out if I don't have a thyroid or something like that. Yeah, it was $120.
Oh, yeah,
it's pricey. I do go to the doctors, yes, because I think it's important. But I always hate to go unless I have, for something like this, I don't want to just show up and go. I want to show up and say, okay, this is what A and B and C and D, and now you need to help me. So I like to have my data with me as a scientist.
Well, even if you went there before you did all that, all they tell you
is to do that. Exactly. Exactly. So I figure I'll go with ammunition and see. Plus it's fun because my husband and I are in competition. He was all mad at me yesterday. How'd you get that many steps in? I can't believe I got that many. You're cheating. It's like, how am I cheating? The thing does what it's supposed to do. It's not like a pedometer. I can't wave my arm like this and get steps. That would be cheating. So we'll see how it goes. So the reason I'm wearing this is because when we talk about nutrition, which was what the beginning of the chapter was, we talk about some of the different components that we put into our body to make us who we are. We have to make certain molecules. We have to use certain molecules to make our proteins. We need to make energy for all the chemistry that goes on in our body. So the second part of the chapter talks about the making of the energy part of the equation with respect to nutrition. This reaction is a very generalized reaction that talks about the making of the energy needed for all of our reactions of metabolism. So when I say metabolism, I should say, what's another word I could use as a synonym?
Energy.
Some, maybe, no. It's one that you probably all hate, and when your teachers say you have to take this course, you get nervous. Phys ed. Those are my two favorite classes in school, gym and lunch. Those are my two favorite. Well, lunch wasn't a class. But yeah, gym and lunch.
It's still broken up as a time period like a class.
That's right. Nobody's got the word? I'll give you the first letter. It begins with a C. Chemistry. Exactly. So metabolism. Yeah, see? I told you you'd cringe. Metabolism, chemistry. Kind of synonyms. So when you talk about metabolic reactions within an organism, you talk about the chemistry that organism has to maintain homeostasis. This chemical reaction we're going to talk about, it's only one of many Did you guys look back at that poster in the lab? Did I mention the poster in the lab to you guys?
No.
When you have lab on Thursday, look on the back wall. There's a poster back there. I don't remember what it's entitled, but it's a pathway of a whole bunch of different metabolic reactions in the body. Scary as hell. So we're going to take a little...
You want us to look at it?
Yeah, just so you get an appreciation for it, to see how complex you are. So we're looking at a little piece of that poster, a reaction called cellular respiration. And that reaction involves 3 major steps. Now again, it depends on the textbook you look at. They can break it down and say it's 4 steps, or some people say it's 5 steps. But our book seems to break it out into 4 distinct steps. The first reaction is called glycolysis, where we start the reaction with breaking down carbohydrates. Simple carbohydrates, like glucose, for example, is the most efficient molecule to use in this reaction. Glycolysis is the what? Yeah, breakdown of sugar. So glycolysis ends with the formation of a very important molecule that's going to continue the reaction going. And the reaction is kind of like a series of dominoes falling. When I hit the first one, I'm going to start the series. And all of these reactions are going to be like a series of chemical reactions with an end product of making energy, ATP. We make an important molecule that's going to continue the series of reactions called pyruvic acid at the end of glycolysis. With pyruvic acid, I'm going to make a very important enzyme molecule called acetyl-CoA, acetyl-coenzyme A. That's important for the next part of the reaction. And the next part of the reaction is a series of chemical reactions we're going to call the Krebs cycle. We're going to make a whole bunch of different things in these previous reactions to bring to the last part of the reaction. The last part of the reaction is where I'm going to make most of my ATP. That's called oxidative phosphorylation, or the electron transport chain.
Oxidative what?
Phosphorylation. See, it's right up there on the little...
And that's where most of what is.
That's where most of my ATP is made. So it's broken down into 3 stages based on the reactions that are taking place. The first stage is where we bring in the stuff that we're going to need for metabolism, the building blocks. What are some of the things that we discussed in nutrition? Those building blocks. So the building blocks I use for this reaction are those calorie molecules, the proteins. Proteins were made of what? Amino acids. So I can use those amino acids in this reaction. The other calorie molecules were carbohydrates, simple sugars. Those guys are in the middle of this diagram because those guys are the most efficient ones to use in this reaction. They're going to give us the most ATP at the end. And then the third one we can use, the other calorie molecule we can use in this reaction, is lipids. And lipids were made of what? Yep, fatty acids and glycerol. So this is a nice diagram because it shows you all the little components and how they fit into this major reaction of cellular respiration. So I can use, again, smack dab in the middle, glucose and other simple sugars. Those are the ones that are going to give me the biggest bang for my buck. But I can also use amino acids, and I can also use glycerol and fatty acids.
Can you explain Krebs cycle again?
The Krebs cycle is a series of chemical reactions in cellular respiration. And basically what that reaction, that part of the reaction is going to do is it's going to give us components we're going to need for the last part of the reaction. As I go along, it'll make more sense.
I'm sorry, I'm late. I had an
No
accident.
problem. Oh, jeez.
So I'm really sorry.
No problem.
Can you please, because I know that you probably went through that, but what do we need to talk about for this diagram?
I'm going to tell you as we go along. Okay, so from this diagram, everything.
Everything.
Yeah, because this is a very basic outline of cellular respiration. So you need to know that there's 3 stages. You need to know that it involves reactions that pull things apart and put things together. And you need to know that what feeds into the reaction are those calorie molecules at the top of the diagram. So we are now going to describe each of them. So
Do we need to know all the chemicals that involve?
no, absolutely not. So the basis, and that's what's nice about this diagram, is it's the basis. Now, oh yeah, okay, this was part of the nutrition part of the discussion. But minerals that are required for other reactions of metabolism are listed here. Again, I'm not going to make you memorize them all, but understand that they're important for building the molecules we need to build us. So, calcium, phosphorus, and those are things we've heard of before, right? Yeah, bones, calcium, phosphorus, sulfur, potassium. All these guys, even though we only need very little amounts of them, are extremely important for many reactions of metabolism. And what did I say the word we could use in place of metabolism was? Chemistry, okay? So that's a list of some of the important guys used for chemistry. So we defined in last class oxidation and reduction, yes? And we defined in last class anabolism and catabolism, right? So when we talk about this reaction, it's a combination of breaking things apart and putting things together. And the goal is to make ATP. So in any metabolism, when I discuss chemistry or metabolism in the human system, one of the important factors is to use as little energy as possible to create whatever reaction it is we're talking about. And that's where enzymes come into play. So figure 24.4 on page 916 in your textbook gives us a visual of some of the enzymes that are important in the end product, ATP. ATP, and we wrote this on the board, was adenosine triphosphate, yes? And we take phosphates in the oxidative phosphorylation part of the reaction and tack them on to ADP molecules. So this little diagram shows us some of the proteins involved in the phosphorylation process, the end product, which is going to help give us lots and lots of ATP. That's why the last reaction is sometimes called oxidative phosphorylation, because that's exactly what we're doing in the end, is phosphorylating a lot of ADPs with the help of ions that are carried to these proteins in the plasma membrane of the mitochondria to push these pumps, drive these pumps, so that we can make more ATP. Okay?
But how about Glycolysis and Krebs Cycle? They also make some ATP.
Correct.
Both.
Correct. Yes.
Each.
Yeah. Lots of
So
reactions.
Do you have this
Nope.
ATPase?
Nope.
No? But,
Nope.
But how they
Is this the only way to phosphorylate something?
No, we can add
No. You learned back, and I'll give you an example. You learned back in when you discussed muscles. I hope you learned 3 different ways to make energy. You had 2 ways that were anaerobic and 1 that was aerobic, correct? So this is not the only way to make ATP. Glycolysis can make ATP. Krebs cycle can make ATP. Direct phosphorylation can make ATP. So those ways are also involved in the production of ATP.
So what's the basic definition of phosphorylation?
Adding a phosphate. Adding a phosphate. Yep. And it doesn't have to be to ADP either. It can be to anything. So we phosphorylate, as, for example, when we begin the process of glycolysis, and I'll throw out some unnecessary chemistry, we phosphorylate the glucose molecule. The reason we do that is we make it just a little bit bigger so it can't bounce back out of the cell. It's called glucose 6-phosphate. So we take a phosphate and we stick it on the 6th carbon of our glucose molecule, and that makes it just big enough so it doesn't bounce back out of the cell. And then we can start the reaction. So phosphorylation happens all the time in many different reactions of metabolism. So enzymes, these enzymes are the ones that are going to help make most of the ATP by doing most of the reaction at the end, oxidative phosphorylation. So again, we look at this in another way, and we see all the different reactions involved in cellular respiration. Again, all these steps build on top of each other. One of the things I use as an example is, this is like a little factory. Its goal is to make energy. So if I, for example, go to a teddy bear factory, I have different departments at the teddy bear factory, right? One department makes eyeballs, and one department makes the head, and another department makes the body. Yes? And all the departments are necessary in order to make the end product, the teddy bear, right? Same thing with this reaction. All of these different reactions are going to produce something that will then help the next part of the reaction take place. Some of them can produce small amounts of energy on their own, but you're a human being. What kind of organism are you? I'm going way, way back. I don't know if you've ever even heard this one. Hopefully you have. You're a eukaryotic organism. Do you know what that means? It's something about your cells, exactly. You have big honkin' cells. You have big honkin' cells with lots of organelles that are membrane bound and do what? Take a lot of energy in order to do their job, okay? Some of them multiply, some of them reproduce. So you need a lot of energy to be a human being. We can make small amounts with some of these reactions. Say, for example, if I'm a yeast or I'm a bacteria, I might actually just use glycolysis to make the energy I need. But you need a lot more than that. So you need a reaction or series of reactions. It's going to give you all of the energy you need to do all of the work that you need as humans. So the first step, again, glycolysis. It's the breaking down of glucose. That's going to produce pyruvic acid. Pyruvic acid, just like at the teddy bear factory, the guy that makes the eyeballs, then is going to go to the next guy, to the head portion, and give my eyeballs to the head guy, or give my pyruvic acid to make another very important molecule called acetyl coenzyme A. Acetyl coenzyme A is then going to feed into the Krebs cycle. And through both of these reactions, glycolysis and the Krebs cycle, we're going to make what we call electron carriers. There's two different kinds of electron carriers that are produced in this type of cellular respiration. One of them is called NAD, and one of them is called FAD. Both of these guys are going to be involved in carrying electrons, hydrogen ions, to the last part of the reaction, which is also called what? Besides oxidative phosphorylation, it's also called the electron transport chain. So through the transport of electrons through those proteins that we saw in the previous diagram, I'm going to be able to carry on oxidative phosphorylation. Do you see the connection there?
Can you again repeat this? It's so complicated. Just step by step.
Yeah, why don't, no, because what I'm going to do now is go step by step and talk about each of them, and then show you where they come in and where they dump their product. So now, what I don't want you to know, and what I don't want you to get caught up about, is the details of each of these reactions. because they're much more complex than we need to cover in this class. When you take your biochemistry class, did I already tell you this story about my biochemistry class in college? Do you know what one of my questions was on my biochemistry final? One question. I had to draw this entire reaction on a piece of poster board. I'm not kidding. Crazy. That was only one question on my test.
Oh.
Yeah.
I thought that was your only question.
No.
And how many more
But
questions?
still, to
I
draw
don't
that on a
remember.
poster board with
Yeah.
How long did you have for this test? Yeah.
But I'm not going to make you do that.
Okay.
Basically, what I want you to understand is the reaction's purpose and the outcome. What does the reaction give me to go on to the next part of the reaction? So the book breaks it down specifics, okay? It breaks it down into the exact chemical reactions that go on during glycolysis. Again, I don't want you to memorize that, but do get an appreciation for how complex it is. What you see at the end is what I want you to know of glycolysis. So we start with a glucose molecule. Then we end with some byproducts that we're going to use. When you look at chemistry and chemical reactions, you see these little arrows with things coming in and things going out. Do you see those little things in the diagram?
Yeah. But where are they going?
They're used to create the next part of the reaction.
No, I mean, what's this arrow? It's showing that, like, for example, phase 3, 4 ADP, and what does it mean? It go out or go in?
No, it's going to result in what?
4 ATP.
For ATP after this reaction takes place. Yes?
And then it will create 2 coenzyme with hydrogen.
Correct.
This is what, do we need to know, like, fructose?
No.
or-
No. No. No, Mila, no.
So all you need to know is that
So you need to know use it.
ATP to ADP.
Well, glucose, this reaction, glycolysis, is going to take glucose, and it's going to actually, you know what? If you look on page, let me give you the final breakdown, so you don't get nervous. It's on page 924, and it's figure 24.12. So glycolysis is a series of reactions that's going to give us two things. Well, that's a waste product that I want to mention before I go on, but it's going to give us two NADs, and let me flip to that diagram, 2412. There it is. Correct. So after the series of reactions takes place and things come in and things go out, we're going to have a net of 2 ATPs. We actually make 4 ATPs, but we use 2 ATPs for the reaction to take place.
So we need to know that. So we actually.
You need to know that. Yes?
Yes.
So the net result with respect to ATP from glycolysis is 2 ATPs. The other thing glycolysis gives us is these electron carriers that I mentioned before. We get 2 of them from glycolysis. These guys are going to pick up electrons from the area and shuttle them over to the electron transport chain. So we get 2 NADH plus H from glycolysis. The other thing we get is pyruvic acid. So pyruvic acid is necessary to make another very important molecule in the mitochondria. So in the mitochondria, I'm going to use pyruvic acid to create a molecule called acetyl-CoA. I actually make 2 of them. In the process of converting pyruvic acid to acetyl-CoA, I'm going to make a couple more of my electron carriers as well. So we see another 2 NAD H plus H.
So in the process of getting the other acetyl, where you get 2 more, that's all before the Krebs?
Correct. So I take pyruvic acid and I alter it. In some textbooks, they'll define it as a separate step. In this textbook, they don't seem to define it as a separate step. But it depends on the writer and depends on the, how it's being presented. But the formation of acetyl coenzyme A is extremely important for the next part of the reaction.
So it's a little bit confusing. So from glycolysis, how many coenzymes will we have after glycolysis?
2.
But why this arrow goes to the Krebs cycle and it says 2 as well? So like 2, you see like 2 different arrows. One
Well, that's
is
not-
above and one is
Because you also, from this reaction to that reaction, are going to create what?
2 NAD
2 NADHs.
H's plus the 2 acetyl-CoA.
Correct.
so it's not from glycolysis. It's
This is from glycolysis. That 2 NADs is from conversion of pyruvic acid to acetyl-CoA.
Okay, from pyruvic acid.
Yeah, that's why the arrow's above that arrow.
And then from the Krebs cycle, you get 6 NADH.
Correct. Now, during the Krebs cycle, which is again a series of breakdowns and alterations, you're going to get 6 NADHs plus H. Again, these are our electron carriers, and a different one, which we call FAD. So NAD, FAD, electron carriers. When you see the H's, what are they doing? Carrying electrons. What's hydrogen? What's a positive hydrogen ion? That's a proton. Pretty much.
A proton is no electron.
Right, but that's what's confusing. It's a charged particle.
A proton is no electrons or more it doesn't have more protons than electrons.
No, just think it's a charged particle.
It's just a nucleus with no electrons. Right?
Pretty much. So that's what it's carrying, hydrogen ions. Okay? So those ion carriers, let's call them ion carriers instead so it doesn't confuse us. Okay? Is that better? So those ion carriers are what we need to bring ions to the last part of the reaction, which is the electron transport chain. After the Krebs cycle, we're going to get a little more ATP. How much? About 2. But a lot of those ATPs that we get are what we call substrate level phosphorylation ATPs. You learned this when you talked muscles last semester, right? Did those last a long time? If I just took ADP and I stuck it on there with these reactions, did those ATP molecules last a long time? No. When I add oxygen to the mix and I pump those ions through the electron transport chain, that's going to give me really strong bonds, ATP molecules, that are going to last a lot longer than quickie substrate level phosphorylated ATPs. So from the electron transport chain, with the help of what? What do we need to perform this series of chemical reactions in the electron transport chain? Electrons, right? Where are they coming from? Or ions, where are they coming from? I wouldn't be able to get them unless, no, what happened? Unless I got them from glycolysis, which ultimately would be sugar, right? But it's not always sugar, it could be other things. Glycolysis and the Krebs cycle. Okay, so that's where I'm getting these ion carriers.
Can I ask you why it's 2FADH2, not plus H?
It's the chemical makeup of that molecule.
But it's the same thing.
Correct. It's bringing the H's to the electron transport chain. Okay? So this is the basis of what you need to know from this reaction. The chapter is going to go into details, how this happens.
He wants to bypass all that.
Yeah, I'm not going to get crazy about it. Just a quick go over it.
Use that phrase a lot. I'm not going to get crazy.
No, well, I'm not going to make you memorize all the details of it. There's no need for it.
I know.
So this is the bottom line. I have to get those ions to that pump. That's the electron transport chain. And you can see them, little hydrogen ions being pumped through the membrane and those pumps. They actually physically cause them to spin. Science has been able to actually see them, where are we? See them under the microscope, electron microscope, of course. But these are actual little rotator proteins, and we can see them on the mitochondrial membranes. Okay? So, this is the Krebs cycle. Have an appreciation for it. Again, pyruvic acid, we're going to convert to acetyl coenzyme A, and then through a series of chemical reactions, we are going to get some ATP. We are going to get our electron carriers. We also, in this reaction, don't forget, are going to make some waste products. What are the waste products of cellular respiration? Heat's one, but that's not necessarily a waste product. It also helps.
Because we don't use,
You don't use it?
No, we use it, but it's not, it's more like we're losing the,
No, we use it for reactions of metabolism. We use it to speed up molecules for reactions of
it
metabolism.
says that it's something that, how is it called?
They calls it a waste product, but it's not always, that's not literal. Waste product from photosynthesis is oxygen. Exactly, because who else uses it? We use it. Not only do we use it, but the plant uses it too. So even though it's not necessary for that reaction, and that's basically what they're saying with waste product, it's something we use somewhere else. Okay? One we don't use from this reaction. Anybody know? We use water, we use the heat. One of them is carbon dioxide.
Yeah, we just,
And that's going to be, that's where we're going to, yeah, exactly. We're going to take that out of our system and release it into the atmosphere. So this is oxidative phosphorylation, where we're pumping those electrons through these little proteins that are on the surface of the mitochondrial membrane. And as we push those protons or push those ions through these proteins, we create enough energy to phosphorylate, oxidatively phosphorylate ADP into ATP. Now these guys at the end of this little complex complex 5 called ATP synthase, at the end these molecules are going to be much more hardy. They're going to last longer and be much more useful for our eukaryotic cells. Again, in your textbook we go through some of the different electron energy gradients as they are transported through the electron transport chain, and we actually see that little ATP synthase rotator ring structure that we see in the illustrations. We can actually see them under the microscope.
You're looking at raspberry.
Oh yeah, at the top of a raspberry?
Top of it, yeah.
So this is the illustration of what that rotator ring is like. So it actually physically causes it to rotate when ions are pumped through it. I'm trying to think if mastering has a nice little, there's a bioflick.
there's no videos.
No, it's not for interactive physiology. It's a bioflick. I have to go and check and see if I can maybe send you a link to it. but it shows the molecular workings of cellular respiration. So this is the bottom line, okay guys? On page 924, figure 24.12. Again, I want you to understand the different parts of the reactions. I want you to know what goes in, what comes out. I want you to know how much ATP is the result of this reaction. And then I want you to also not forget the electron carriers that are produced from the series as well.
So if that would be one of the possible say questions, how we can write a say about that? How would you like us?
If I just the way I...
Order you want us to, because for me it's hard. Like I can draw this, but just make it as they say. I don't know from what I would start, like from glucolysis, and then I'm gonna explain.
How would you, how did I draw it out for you when I explained it to you?
Yeah, but you said that you don't need us to
No.
know the details, so that's why I'm.
Yeah, but when I explained it to you, I explained all the steps, I explained what came out, I explained what went in. So that's how you would explain it to me if you had to teach it to me, right? And if that's how you would explain it to me if you had to teach it to me, then that's how you would write it in an essay. You could use a drawing too to help you out.
Yeah, but my question, because you said that you don't have to memorize the details, but when
Correct.
you explained, you actually give us some details. That's why I'm asking.
Okay. Explain that diagram to me. Would be how you would answer that essay question. Yes?
I think
Okay.
what she's asking is like, where do you start, where do you end?
Well, where does the diagram start?
Start with glycolysis.
Yeah. So that would be a reasonable place to start.
And you, from there, well, I'm a little confused. So the arrow that goes down from glycolysis, you automatically get 2 ATPs.
Remember, you have to go through that big bunch of reactions. But the result is going to be things that come out of the arrows.
But to go up, you have, when you go up, you get your 2 NADHs plus H, which go into the electron transport chain.
Mm-hmm.
And the result
So
of that,
remember the teddy bear factory?
yes.
The guys making eyeballs make eyeballs.
So the result of that is the 28.
Right, but then the eyeballs go to somebody else, somewhere else. Yes?
Right.
So the result of glycolysis would be a couple of things. One would be ATP, and another thing that is a result of that series of chemical reaction.
2 NADH plus H.
Correct.
Can we make 4 of them now? No. Because I'm looking at 2 on the top. It does, doesn't it? You
Yeah,
just don't want
just
to tell me.
don't get confused by that.
Okay.
What I want you to understand is that's what comes out of the reaction. Okay? It actually, if you want to get confused, it takes 2 turns for each glucose molecule, so you double everything.
So
We don't need to know that.
I agree.
Right?
All you want to do, Mila, is show a reasonable understanding.
Right.
I mean,
So
she's not
basically,
hard on the essays.
right. No. At least I didn't think I was. But anyway, what I want you to understand is the arrows and what they mean. So things coming in and things coming out. There's a whole bunch of stuff in between that I don't need to know about. Okay?
after the Krebs cycle, I just want to clarify.
Yep, go ahead.
After the Krebs cycle,
So what do we get from the Krebs cycle?
you get 2 ATPs.
Correct.
And you also get 6 NADHs and 2 FADHs.
Correct.
Okay. I just want to make sure that I'm like reading it
Yep.
correctly.
But we
And
also
then
have
what-
to remember that the pyruvic acid is when it's made into the acetyl
Right.
CoA. Before that, it also makes 2 more NADH plus H in the process of making the acetyl.
Correct. So do you see what she said in words? Okay. So it would be basically explaining where the arrows come from. So from pyruvic acid to acetyl coenzyme A, and pyruvic acid is formed after what? After glycolysis, we also create 2 more what?
NADH.
Correct.
Just remember a couple of
No, sir. I want to make sure the words and numbers are in order, Anthony. Right?
It's all about the effort.
Right. But that's basically what this diagram is showing us. So don't get confused. And again, I'm having a hard time seeing the confusion. But
I think it's just because it's not like it goes right. There's like all these different.
see, but it's all over the place until you understand what's going on, and that's my job to explain to you what's going on.
I think what it is like you have something here and it goes this way and this way.
Right.
And then this way goes on this way and this way.
And that's why I keep using the teddy bear factory as an example. And I know you're going to kick me in the head if I say it one more time. But if my job is to make the eyeballs, that's what I do. I make the eyeballs. Okay? Okay, let's give that department 2 jobs. Eyeballs and legs. Eyeballs and legs. Okay? Now the eyeballs are going to go to the next guy. And the legs are going to go to the next guy. But he keeps making eyeballs and legs. Glycolysis keeps making.
I understand
Yeah?
bears. I don't understand chemistry.
Yeah, but I'm trying to make it not so scary. So glycolysis makes 2 things. Correct. Now what we might use the ATP.
Makes 2 things. So glycolysis, the guy makes eyeballs. So then you get the 2 eyeballs, makes 2 things. Like I need association. I know it sounds dumb, but if we're talking about teddy bears and we're talking about chemistry, and none of it makes any damn sense. And right now I'm seeing Kreb Cycle, and I keep thinking about SpongeBob SquarePants.
But why does it not make sense? Because it's just that simple. In order for me to make the teddy bear, and what's the teddy bear?
The ATP.
All the ATP at the end. I have to have these certain things to assemble. In order to make enough ATP to survive as a eukaryotic organism, I need all of these other building blocks so I can make enough.
So that bottom line is just the ATP. It's
Yep.
just what
And the top is,
you end up with, right?
correct.
That's what you end up with.
And the top part is the electron carriers that are going to help me the end product, exactly. And again, some reactions will give me both. So that's why you see the arrows coming from the top and arrows coming from the bottom. Yeah, from 1 glucose molecule. Actually, it's how much?
Well, 30 total it says, but I think that's.
Yeah, 30 to 32.
Though it's 32, but
Correct.
I think it's because that one says about.
Correct.
28.
And it depends. So some of them get used up quicker. And again, you can't be totally exact all the time in chemistry. You might use more, you might use less. So if you read a different textbook, not to confuse the hell out of you, but you'll get different numbers for this as well.
You could have a faulty glucose molecule to start with.
Or you could use up more energy for a reaction because of other reactions of metabolism going on.
You don't have enough stuffing. I don't even know. I'm ready to just like.
Why is that confusing you? It's an analogy. Is every single molecule going to be the same every time?
No, but I guess I might.
Okay, so nix the teddy bears and stick to the chemistry.
I just wish it was listed from top to bottom.
It is. It's listed from side to side. Turn it this way and it'll be top to bottom.
So I guess what I need to do is
Correct.
literally just do that. And
Yep.
then I'll be okay, because this is not a good diagram. I think it's junk. But
Okay, so take that diagram and turn it this way and it'll be from top to bottom.
okay.
Yep.
So that's my problem. So I'll do that when I.
Well, that's easy. That's an easy fix.
I'm gonna do that.
Good. Okay. So, now to complicate things a little more, we don't necessarily need glucose to do it. So glucose is optimal. Glucose is going to give us the optimal 32 ATPs at the end. But we can use other molecules. Yeah, we can use fat. We can use glycerol and fatty acids. And we can use, what else? What was the other calorie molecule? Well, carbs is glucose. Yep, say it. Proteins, right? Amino acids. So the next part of the book talks about glycogenesis, glycogenolysis, and gluconeogenesis.
Yeah, those are some nice words.
Aren't they awesome? But they all have gluco in front of them. So they all imply some way that they're somehow linked. So making glucose, glycogenesis, breaking down glucose, glycogenolysis, and making new glucose, gluconeogenesis, are the basis of each of these terms. So we already talked about breaking down glucose, right? We stick it in glycolysis, we break down glucose, we run through the reaction as said. Now, again, that's the best way to do it. This is where we're going to get the highest yield, ATP, and that's what they describe under glycogenesis. Glycolysis is breaking down that molecule in the reaction of cellular respiration. Lyse means to what? To break apart. So in this reaction, we're using glucose that we've built, we're bringing it into the cell, and then we're altering it. And we see that, remember that molecule I talked about, glucose 6-phosphate? That's how we keep it in the cell so that we can start breaking it down. Carbohydrates are necessary for energy production. The more energy production I have, the more reactions of metabolism can take place. So they talk a little bit about athletes and carbohydrates. Why are carbohydrates important for athletic?
Slow energy release.
Well, actually, well, complex carbohydrates are slower, right? If I have a simple sugar, I have a higher energy release. But again, when I'm an athlete, I'm performing muscular activity, I'm going to need more energy to do that. So carbohydrates are important. Because I don't want to use them up all too fast, I have the ability to store them in places. In the form of what? And you've heard this one before.
What are we storing?
Glucose. Glycogen. Okay, where do we store glycogen? Places like the liver, in our muscles, okay? So we store some of those carbohydrates, but then in order to use them, and that's what they talk about under glycogenesis, in order to use them, what do we have to do? Break them down. Glyco, exactly. Exactly. And that's glycogenolysis, breaking down of who? Glycogen. And that's going to release the glucose molecules that I need. Now, I told you you could use other things, and that's what this little diagram, by the way, shows us, is the making and the breaking of glycogen. So when I bring in glucose molecules, I can use some of them right away to make energy, and I can store some of them. I can store some of them in the form of glycogen. What if I have even more than that? No, I am very efficient. I turn it into what? I turn it into lipids. I store it in the form of long-term storage with just fat. So, why?
Why not protein?
Well, because it's not an amino acid. If it was extra amino acids, we'd talk, but it's not. Sorry. So, initial phase of lipid oxidation. I can break down lipids for energy. So the first one they talk about, the new glucose, making new glucose, gluconeogenesis, is breaking down these other molecules to feed into the chemical reaction of cellular respiration. And I'm not going to say anything about the teddy bear factory, but remember the end product. We don't necessarily have to have all of the different things in order. It's the end product that we're concerned with. So I can use lipids. We can use glycerol and we can use fatty acids. And that's what they discuss under lipid metabolism.
So they use both?
Yep. So part of the breakdown, a series of chemical reactions of the fatty acids of a lipid molecule can be used to make acetyl coenzyme A. And now we know what we're going to do. What are we going to do with acetyl coenzyme A? We're going to bring it to the Krebs cycle.
And the Krebs cycle creates the 6 NADH and the 2 FAD.
a little bit of energy, and then those ions are going to be carried where?
To the transport chain.
Correct. So we won't make as much, but we'll still make some really good, healthy ATP at the end.
So you said we can use lipids, fatty acids, and what was the third one?
Well, let's get through lipids, and then we'll go to the next one.
Okay, sorry.
Okay? So we can use glycerol as well. Alter it a little bit. It can actually be then fed into glycolysis to continue on its way. Pyruvic acid, acetyl coenzyme A. Again, I'm not going to make you memorize the details of these reactions. I just want you to understand that fatty acids and glycerol can be used to sort of feed into a reaction of cellular respiration as well.
You know, like, for example, glycerol will transfer to chemical that will go to the
No.
glucose.
You just know that glycerol can be used fed into glycolysis, and fatty acids can be used to make acetyl coenzyme A. This is the actual chemical breakdown when we use lipids to make our energy. One of the things that I want to point out here, again, I'm not going to get crazy about the reaction. I'm not going to get crazy about the specifics. But I do want to point this out, that one of the downfalls of using this as our energy source too fast is that we create waste products that our body then has to filter out. And who's responsible for filtering out the waste product produced by breaking down lipids, gluconeogenesis, ketones, and the liver is responsible for filtering that out.
So what's the
Ketones is an acidic compound that is a waste product.
And those do show in your
Correct.
Can I ask you, so that waste product of
That's a
that
true
glycerol
waste product. That's a true waste product
of both glycerol and
of fatty acid metabolism,
okay, fatty
lipid metabolism. Okay?
So the waste is the ketone bodies.
Correct. Now, ketone bodies are very acidic. And what can they do to our pH? No. Lower it. As acid. Remember the definition of it? Yeah. Alkaline would
I
be,
always forget acidic.
yeah. Hydrogen ions in solution, right? So pH, the more hydrogen ions we have in solution, the more acidic the solution is. And the lower the what? That's, yeah, confusion. So that's one of the downfalls of using lipids, the majority of our energy-making molecule as lipids, or trying to use lipids too fast. That's one of the big problems with a diet, for example, that causes you to burn lipids too fast. You produce these waste products that your body then has to deal with, and they can disrupt homeostasis when we use them too much. Now, it's not bad to use lipids. As a matter of fact, your skeletal muscle likes lipids. It uses it, no problem. But it uses it at a nice even rate so that your body, your liver, can filter out the ketone bodies and your urinary system can get rid of them for you in a nice even rate. Also, acetyl coenzyme A, not to confuse you, but I just pointed out because it's in there, it's also used in the production of cholesterol too. So not only in this reaction of cellular respiration, but it's also used to make cholesterol as well. Acetyl coenzyme A.
Is it kind of like a waste product as well?
No, not cholesterol. What are you going to do with your cholesterol?
No, but go back to, can you show this diagram? You see like sort of
Acetyl coenzyme A.
enzyme will go to the Krebs cycle, right?
Yep.
And the same acetyl coenzyme will make a cholesterol.
Correct. Will help to make
All
cholesterol.
that.
Yep. And what do you do with cholesterol?
Everything.
You put it, it's, yeah, exactly. And you also use it to make hormones, and see the arrows? And you also use it to make bile salts, yes? Yes? Are we becoming one with the arrows a little bit more? So this is going to lead to this as well. Okay? Yes?
If you can't answer this quickly, it's my fault because I don't want to answer it because I missed the class. Catabolic and anabolic, I noticed that it's reactions kind of reversing direction in some places.
Yep.
So can you just give me
Catabolic would be a reaction that breaks things apart.
Oh,
Anabolic would be a reaction that brings things together. So again, there's different things that are happening in these reactions.
It's actually, it's a catabolic, so it's kind of helps the role
So
break
a breaking down of this will lead to this. This as a component, along with other things, will lead to that. Make sense? And I'm not going to make you memorize that. That would be cruel and inhuman. All right? So, this is the liver and its role in breaking down some molecules, using some of their components, and then getting rid of waste products. So, keto acids are modified by the liver and then put into the Krebs cycle. But keto acids are also a waste product as well. So we can actually use some of the keto acids in the Krebs cycle, but the rest, the waste products, need to be removed by the urinary system. What we'll do with the... well, it's about... Yeah, but I'm talking about the ketone bodies as well. So...
Who says that the keto acid and keto bodies is absolutely different things,
Correct.
right?
Yes.
I just want to make sure because I'm confused now.
Yep. No, keto acids from amino acid metabolism are dealt with in a different way. So we can use some of them in the Krebs cycle, and some of them are going to be altered by the liver and produce waste products. Carbon dioxide and what else? Urea. Ammonia and urea are some of the waste products of using amino acids as our energy source. Do we want to use amino acids to make ATP? No. What do we want to do with our amino acids?
Building blocks for our
Yeah, we want to make our proteins. We have so many to make. Muscle, antibodies, enzymes. We don't want to use them to make energy. Why would we use them to make energy?
Because we don't have the other
Building blocks.
Right, we don't get their building
So when you starve yourself, what are you going to do?
start using other sources.
Correct. You're going to start breaking down those proteins because you don't have the other building blocks available to make ATP. Your body's protection mechanism, protecting you. Your metabolism is going to go way, way down, and you're going to start breaking down your own proteins to make the energy that you need to survive.
So all the amino acids metabolism will be in the liver, only in the liver, or somewhere else?
Well, not only in the liver, no. Amino acid and ketone body, this is the alteration of some of our amino acids when we break down to use them as energy source. But amino acids are put together all over the body. So in many different cellular. Just think of transcription and translation. Every single cell has the ability to produce a protein. So not just the liver. This is the alteration of amino acids to use for energy molecules. Okay? So that's described under the oxidation of amino acids in your textbook starting on page 928.
So again, this table is
Diagram.
confusing because amino acid plus keto acid, and it's again keto acid plus amino acid. So it's the same thing, but they just switch the places.
Mhm.
Switch the, so what does it mean?
So what happens to the keto acids? They're going to get modified and used where?
Oh,
In the Krebs cycle. And what happens to glutamic acid from amino acid metabolism?
It gets wasted.
Yeah, it gets oxidative deamination. It's a chemical reaction. It's going to be turned into ammonia, and ammonia dissociates into carbon dioxide and urea. Those are waste products. They're going to be eliminated from the body.
yeah, but I thought that amino acid is transferred or switched to be a keto acid, right?
No, it's not switched to be. It's in addition to. It's necessary for the chemical reaction to take place.
So I'm just confused.
When I alter an amino acid to produce glutamic acid, I'm going to produce keto acid. So I'm going to have an altered amino acid plus a keto acid.
I just don't understand why.
When I'm going to say it again, listen to what I just said. When I alter an amino acid to produce glutamic acid, I'm going to produce a keto acid. So I'm going to have now, after I alter amino acids, I'm going to have a keto acid plus an altered amino acid. The amino acid is then going to be transformed and dealt with. I can't use it anymore to make my proteins, so I have to get rid of it. The keto acid, I can use a little bit in the Krebs cycle, okay? But that amino acid, that altered amino acid, I can no longer use to build my proteins.
Can I give you feedback? So keto acid, when it's combined with amino acid, it alters both. Both of them alter because
Correct.
of their combinations. And then
But look at, now pay attention to the different forms. There's a different form of keto acid in the beginning of the reaction.
that's what I'm saying, that they alter their chemical.
Correct. And that process is called transamination.
Okay.
So that's going to alter the keto acid and also the amino acid.
Okay.
So even though we're still calling it the same thing, it's not the same thing.
That's why
That's,
confusion came.
I know, that's what I tried to explain.
Yeah. No. Now I understand. So it's
Yeah.
altered.
So it's a different form. So, and those, again, are going to, some can be used, but most, that waste product has to be eliminated. Okay? Again, we don't want to use these, but we can use them if we're in a pinch. So that's what they talk about under oxidation of amino acids. Protein synthesis is basically what we want to do with our amino acids. So we see all three of these guys feed in somewhere into the formation of ATP, somewhere in these reactions. The beginning of the reaction doesn't take any oxygen. So glycolysis is called what? Anaerobic. Oxidative phosphorylation, we need oxygen for that to happen. What's that called? Aerobic. Where are we going to get more ATP from? Aerobic or anaerobic respiration? Aerobic respiration. What are some of the waste products we have to deal with when we alter molecules that, besides carbohydrates? Too many of them. Proteins, we're going to get, what's a waste product? Urea, exactly. How about lipids? Ketone bodies, exactly. If I just use glycolysis too much, I can also build up a waste product called what? Lactic acid. And lactic acid buildup, anybody ever run for long periods of time? Do you ever get cramps in your legs? Burning sensation in your leg? Yeah, and that's a buildup of lactic acid. What's happening is you're using the oxygen that you're taking into your body at a very rapid rate. So sometimes you have to rely on anaerobic respiration, glycolysis, to make a little more ATP than it normally does. And that waste product builds up in your muscles. It's fermentation is similar to this process. So when we talk about simple organisms and making bread, for example, fermentation process involves glycolysis of yeast. And yeast are going to give off a little bit of energy, but they're also going to give off some of the waste products of fermentation, lactic acid, and what else? CO2. And that's what causes your bread to do what? Rise. It causes your bread to rise. Okay?
I just learned some good time, some pancake science over the weekend.
Pancake science.
It's relevant. Buttermilk is highly acidic, and when you mix buttermilk with baking soda, it makes the bread get fluffier through CO2. Because I forgot to do the baking soda, my pancake was like a heavy
lead bomb.
And my dog loved it. But then I realized what I had done wrong, and I put like a tiny bit of baking soda, and it was like, it was amazing.
Totally different, right? So when you bake, when you bake, forget the baking soda, forget the baking powder, and they have, basically the same kind of thing going on, gas production pretty much. Huh? Put vinegar in? Oh, pancakes?
Vinegar and plus baking soda. They both actually.
Yeah, well, that's how everybody made their volcanoes way back when, right?
And if you don't have buttermilk, you can use a little vinegar in regular milk, and it's the same thing.
Correct.
That's what I had to do.
And there's the chemistry of cooking. Exactly. I think you should bring your samples to class so we can taste test them.
Maria, so Krebs Cycle is anaerobic, right?
Krebs Cycle is, yeah.
Because it makes only 2 ATP.
Yeah, well, it doesn't involve oxygen. I'm trying to look at,
It's actually, the book that says that it's aerobic.
Krebs Cycle?
Yes. It says that they,
Oh yeah, because, yes, it is aerobic.
it's aerobic.
Yep.
Because both of them need
Oxygen.
oxygen, Krebs Cycle.
Yeah, for the formation of
Oxidative phosphorylation.
phosphorylation. So, where are we? We are in fats. And again, now the book gets a little bit repetitive. So it keeps saying the same thing. And it's a whole bunch of different ways. Don't worry about it. Basically showing you where some of our lipids are going to go, how the liver is involved, some of our proteins are going to go. Don't get carried away with that. No. So next thing the book talks about is, or at least in this diagram anyway, insulin's direct nearly all events of absorptive state. Insulin is going to help us do what? You should know that because we already did the endocrine system.
It will help us to uptake glucose.
It's going to help. Correct. It helps get glucose into the cells, right? Increase the rate of absorption of glucose. So when we talk about absorptive state, we're talking about getting glucose into the cell so it can be altered to make ATP. And that's basically all they're talking about here.
Everything is altered to make ATP.
No, not everything. Because you don't want, you need your energy, but, right? You don't want to,
Everything's altered.
well, Are vitamins altered to make ATP? No. Minerals? No. They're involved in other reactions. Everything with calories, let's put it that way. Okay. Are you good with that? You're good with that? All right, so that's what they're talking about, absorptive state. Getting that glucose into the cells in order to manipulate it to make ATP. Again, they go on and on about carbohydrates, triglycerides, amino acids, some of the different hormonal controls. We talked about things like glucagon and insulin with respect to hormonal controls of the absorptive state. So we can break down glycogen to release glucose, and we can use insulin to get glucose into the cells. When plasma glucose levels go way down, we are going to stimulate the release or alteration of other molecules in order to make ATP. So glucagon is a hyperglycemic hormone, which means what? Glucagon is a hypoglycemic hormone. Stimulates. Oh, I see what it means. I see what they're saying. So glucagon is going to help increase blood glucose levels by breaking down glycogen to release glucose.
So why it's saying that it's not only help to decline blood glucose level,
It helps to increase blood glucose levels.
I'm sorry. It's released and stimulated by declining blood glucose.
Correct.
It's also releasing or stimulated by rising amino acid levels. So why it's raising because of amino acid?
Well, what do you have? What do you have to do when amino acids go up in the system?
Break it down.
No.
Where? Inside of cell or outside?
Well, just cellular metabolism is what. When amino acids raise, what do I have to do with them?
Make a protein.
Make a protein. What do I have to do to make a protein? Does that involve energy use? So I have to make more ATP when amino acids raise in the system as well. So when you eat proteins, you have to increase energy production so you can make proteins from the amino acids that you produce. Does that make sense? Yes? What is this one? This one is summary of normal hormonal influences on metabolism. Again, repeat information from your endocrine system chapter. So insulin is going to not only increase uptake of glucose, but what else? Insulin. It's going to what? Yeah, what else?
Glycogenolysis.
Yeah.
Photosynthesis.
No photosynthesis. Lipogenesis, which is what? Fat storage. Because we're very efficient. When we get a lot of stuff in, we're going to make sure we either use it or store it. So insulin, when insulin levels raise very quickly, they're going to allow me to use glucose, but they're also going to take the extra glucose and do what with it? Store it as fat. Exactly. Glucagon, again, is going to stimulate glycogenolysis, breakdown of glycogen release, and that's going to increase glucose levels, stimulate lipolysis and fat metabolism as well. So this is just a general information on some of the different hormones and what they do to blood glucose levels.
Do you want us to know this?
No. You already know it, or you should know it.
All right. We know it.
All right, they also talk about transport, lipid transport. Lipids are hydrophobic molecules. What's that mean? They don't like hanging out in water. They repel water. They're scared of the water. That's right. That's what the phobia means, hydrophobic. So we are going to pick up with cholesterol transport on Thursday of this week. And we'll be very quickly finished with this chapter, so we'll go on to chapter. Yep. So we'll go on to chapter 25, most likely on Thursday as well. Okay?